I want you to just picture something for a second. Imagine holding a tiny, pulsing command center of human emotion, something no bigger than a lentil, and it's just sitting there in a petri dish.
Yeah, it sounds like science fiction, right?
It really does.
Yeah.
But for decades, biologists have basically dismissed nearly half of human DNA as, evolutionary garbage. We called it junk DNA.
Right, the stuff that supposedly didn't do anything.
Exactly. But today, we're looking at a breakthrough study proving that not only is this so-called junk highly active, but it might actually contain the exact molecular switch that causes a mother's stress to permanently rewire her unborn baby's brain. So, welcome to the deep dive. We are unpacking a truly wild piece of scientific literature today.
It's a massive leap forward.
It is, because researchers haven't just grown a human amygdala-like, the brain's main fear and emotion center, in a lab.
Yeah.
They have actually physically wired it to a lab-grown hypothalamus.
Yeah, they essentially built a living, breathing biological model of the human stress circuit.
Okay, let's unpack this from the very beginning. Because how do you even begin to grow a brain region associated with fear outside of a human body?
Well, the stakes here are incredibly high. Understanding this specific circuit is it's critical because the amygdala is at the absolute center of severe neuropsychiatric conditions. We're talking about major depressive disorder, anxiety, PTSD, autism spectrum disorders.
Right, but it's always been super hard to study, hasn't it?
Oh, incredibly hard. Historically, studying the human amygdala has been a massive challenge. It sits deep in the subcortical regions of the brain, so it's largely inaccessible in living humans.
So we usually rely on mice, right?
Yeah, animal models. But the human amygdala possesses completely unique developmental and transcriptional programs. I mean, the actual genetic code running in a human forebrain simply does not exist in rodents.
Wow, okay.
Yeah, so we've been trying to understand this complex human operating system by looking at mammalian hardware that is, millions of years diverged from us.
So we have to build our own. Let's look at the architectural blueprint. If both of these brain regions, the amygdala and the hypothalamus, start as the exact same pluripotent stem cells in a lab, how do you convince one batch of cells to become a fear center and another to become a stress responder?
You basically have to perfectly mimic early human fetal development. And the amygdala is uniquely difficult to grow because it isn't just a uniform lump of tissue.
It's made of different parts.
Exactly. It's a highly complex amalgam that originates from entirely different embryonic zones. During development, it pulls cells from the dorsal regions, which we call the pallial zones, and the ventral regions, known as the subpallial zones.
Okay, so to recreate this in a petri dish, the researchers had to guide these human stem cells through a very specific chemical gauntlet.
Right. They apply a targeted chemical cocktail. So the first step involves a mixture called SFT, which stands for SFRP2, FGF7, and TGF-alpha.
And what does that alphabet soup actually do?
Those signaling proteins act as a localized set of instructions, basically telling the stem cells to differentiate into the ventral pallium. That forms the base layer of our structure.
Got it. The foundation.
But to generate the diverse subnuclei of the amygdala, they need more specific structural elements. So they introduce a retinoid X receptor agonist called SR111237, which drives lateral specification.
Okay.
And finally, they use a compound called promorphamine to activate the sonic hedgehog pathway, or SHH.
I want to focus on that sonic hedgehog pathway because looking at the study, this seems to be where the researchers hit a massive hurdle.
Oh, it was a huge sticking point.
It sounds kind of like building a highly complex cake. SFT is your flour and sugar foundation, SR11237 is your wet ingredients, and sonic hedgehog acts like the baking powder.
I like that analogy.
Right, but if you add the baking powder, the SHH, before the flour has settled, the whole structure just collapses. Timing is literally everything.
That is an excellent way to visualize it. The timing of the SHH pathway activation turned out to be the critical variable. When the researchers applied the sonic hedgehog signal too early, like around day 14 of the cell culture, it severely suppressed two vital genes, EMX1 and EMX2.
And what role do EMX1 and EMX2 play in the final brain structure?
They are crucial transcription factors. And just to clarify, transcription factors are proteins that bind to specific DNA sequences to turn genes on or off.
Right.
In this case, EMX1 and EMX2 turn on the genetic code required to build glutamatergic neurons.
And those are the gas pedals, right? The excitatory ones.
Exactly. These are the excitatory neurons of the brain, the ones that fire action potentials and propagate signals. Without them, your amygdala cannot communicate. It's functionally dead.
Wow. So if the transcription factors are suppressed by early chemicals, the entire excitatory network just fails to boot up.
Right. So the researchers discovered that by utilizing a delayed SHH activation, specifically waiting until between days 16 and 24, they completely bypassed this suppression.
That level of developmental precision is astonishing.
It really is. This precise window allowed the organoid to develop all the deep subpellial characteristics without destroying the architects of the excitatory network.
Okay, but that brings up a huge biological verification problem.
Yeah.
How did they confirm they had actually built an anatomically correct amygdala rather than just, a disorganized ball of brain cells?
Yeah, you have to prove what you built. They turned to a technology called single-cell RNA sequencing.
Okay, I have a way to explain this one. Imagine taking a complex tissue sample and treating it like a fruit smoothie.
Okay.
Traditional sequencing tells you the overall flavor of the smoothie, like, oh, it's berry flavored. But single-cell RNA sequencing allows you to pull the smoothie apart and examine the genetic signature of every individual blueberry, strawberry, and banana.
That's spot on. When they sequenced these human amygdala-like telencephalic organoids, or hATOs, they found a beautifully balanced neural architecture.
Meaning they found the blueberries and the strawberries.
Exactly. The organoids contain both the excitatory glutamatergic neurons we just discussed and the inhibitory GABAergic neurons, the brakes, which are marked by genes like DLX1 and GAD1.
Because you need both the signal propagation and the signal dampening to have a functional neural network.
Right, but having those neurons doesn't guarantee it's an amygdala. It could just be generic forebrain tissue.
So how did they prove the exact address?
To prove its exact zip code in the brain, they searched for overlapping molecular markers. They found a specific nuclear receptor called NR2F2 being co-expressed with a gene called FOXG1.
And what do those tags mean?
In biological terms, FOXG1 acts as a tag that says, I am located in the forebrain, while NR2F2 says, I am specifically localized to the amygdala.
Amazing.
They even identified distinct anatomical subnuclei like the basolateral amygdala and the central amygdala, which were already secreting vital neuropeptides like corticotropin-releasing hormone, or CRH.
Okay, let's do a quick recap so we don't lose the forest for the trees here. I want to deliver a rapid, bulleted summary to just lock this in for you listening. First, the amygdala is uniquely complex to grow because it requires tissue from multiple embryonic zones. Second, precise, delayed chemical timing, specifically the sonic hedgehog pathway, is the secret key. Third, the resulting hATO has both excitatory and inhibitory neural networks.
Perfectly summarized.
So we have a functional mini amygdala, but does it actually communicate, or is it just firing in the dark? Because an amygdala sitting alone in a dish doesn't create a stress response.
Right. It needs a partner.
It needs to talk to the hypothalamus to trigger the body's entire stress cascade, so the researchers had to build a bridge.
Yeah, an isolated amygdala cannot model a systemic stress response. In humans, the amygdala detects a threat and fires a signal to the hypothalamus. The hypothalamus then initiates the HPA axis, the hypothalamic pituitary adrenal axis.
Which is what cascades stress hormones throughout your entire body.
Exactly. To model this, they had to engineer a biological partner, a hypothalamic organoid, or hATO.
Was that as hard to grow as the amygdala?
It had its own challenges. The issue is that standard protocols naturally default to creating the ventral portion of the hypothalamus. But the researchers needed the dorsal portion, specifically a region called the paraventricular nucleus, or PVN.
Why is the PVN so important?
The PVN is the exact anatomical receiver dish for the amygdala stress signals. To force the stem cells to become the PVN, they modified the standard recipe by introducing a dorsalizing morphogen called BMP4.
Okay, so they have a fully formed green amygdala because they tagged it with a green fluorescent protein, right?
Yes, GFP.
And a fully formed red hypothalamus tagged with TD tomato. How do they actually get them to communicate? Do they just mash them together?
Essentially, yes. They call it an assembloid merge. On day 25 of development, they place them in direct physical contact. And over the next 30 days, without any external guidance, reciprocal axons, the long, thread-like wires of the nerve cells, began growing out of the green amygdala and plunging deep into the red hypothalamus and vice versa.
They autonomously wired themselves together.
Yes, they formed a physical circuit.
But okay, physical wires don't guarantee that actual data is transmitting.
Right, you have to test the wire. So they utilized optogenetics. They introduced a light-sensitive protein called crimson R into the green amygdala.
And what does that do?
Crimson R is a specialized ion channel that forcefully opens and fires the neuron when exposed to orange light. Simultaneously, they infected the red hypothalamus with a calcium sensor called GCAM-P6s.
So when a neuron fires, calcium rushes in, and that sensor makes it glow green under a microscope.
Exactly. So they pulsed the orange light onto the amygdala, and immediately the hypothalamus lit up with calcium transients.
The signal successfully crossed the bridge.
It did.
But okay, here's where it gets really interesting. I have to push back a little here. Using a light-gated ion channel to force a reaction, prove the plumbing is connected, sure. But can this engineered circuit actually respond to a biological stressor? Like, does it react the way a human baby in the womb reacts to its mother's stress?
That is the exact right question to ask. And the researchers share that skepticism. Optogenetics proves the physical wiring, but the next step was testing the chemistry. They wanted to directly mimic the biological mechanism of prenatal stress, where elevated maternal cortisol crosses the placental barrier.
So imagine a clinical case where a pregnant mother experiences severe trauma or chronic anxiety. Her body floods with cortisol.
Right, and that cortisol reaches the fetal environment.
What is the actual mechanism when that cortisol hits these fetal brain cells?
Well, the lab-grown amygdalas organically developed glucocorticoid and mineralocorticoid receptors. These proteins normally sit inactive in the cytoplasm of the cell. But when the researchers introduced a physiologically relevant dose of cortisol 10 micromolar for 24 hours, the cortisol molecules physically bound to these receptors.
And then what happens?
This binding causes a structural change, allowing the whole receptor hormone complex to travel directly into the nucleus of the cell. Once inside, it acts as a transcription factor, fundamentally altering gene expression.
So the cortisol acts like a key that literally unlocks the cell's nucleus and starts rewriting its daily instructions.
Exactly the mechanism. And the physical result was profound. It triggered immediate oxidative stress, upregulated stress genes like MT2A and FKBP5, and most importantly, the amygdala started aggressively secreting CRH.
That's the chemical panic signal. But how do we know the amygdala is actually instructing the hypothalamus to react? If they are sitting in the same dish of cortisol, couldn't the hypothalamus just be reacting to the cortisol on its own?
Proving the directionality of the circuit was maybe the most elegant part of the whole study. They utilized a custom microfluidic chip.
Okay, let me visualize this for you listening. Think of this microfluidic chip like two completely sealed, locked rooms. You put the green amygdala in room A and the red hypothalamus in room B. The rooms are connected by a tunnel so narrow that fluid cannot pass through.
Right, so the chemicals can't mix.
Exactly. But the microscopic neural wires, the axons, are small enough to grow through that tunnel and connect the two rooms. It functions exactly like two people with a string telephone behind closed doors. You whisper the stress hormone into the amygdala room, and what happens?
The hypothalamus room reacts perfectly. They observed a massive spike of CRH in the hypothalamus chamber. The stress signal was translated and sent down the physical axons.
But what if you yell cortisol directly into the hypothalamus room?
Absolutely nothing. The hypothalamus chamber showed no significant CRH response. It is functionally deaf to the raw cortisol. It absolutely requires the synaptic translation from the amygdala.
Okay, that is mind-blowing. Let's do our second quick recap here. Bullet 1. The two organoids successfully form a biological circuit. Bullet 2. The amygdala translates raw cortisol into CRH. Bullet 3. The signal travels down the wire to activate the hypothalamus, which can't process the stress on its own.
Perfect.
But that leaves a massive question. We can see the stress circuit firing perfectly, but what is this stress hormone actually damaging inside the cells on a genetic level?
That is where they dug deeper into the RNA sequencing. And they found a hidden, highly surprising mechanism hiding in what we used to call junk DNA.
Oh, this brings us right back to the beginning. Junk DNA. What exactly did they find?
They found that the damage was being orchestrated by transposable elements, often referred to as retrotransposons or jumping genes. These make up about 40% of our entire genome.
40%.
Yeah. Many of them are essentially ancient viral remnants that integrated into our ancestors' DNA millions of years ago. Normally, our cells keep these elements tightly silenced. But the cortisol exposure specifically and massively awakened one particular non-coding RNA retrotransposon known as BCY-RN1, also called BC200.
Why is BCY-RN1 so dangerous when it wakes up?
It comes down to its physical structure. BCY-RN1 contains something called a 5' ALU domain. In the developing brain, its normal function is to regulate translation at the synapses by interacting with a specific RNA helicase called EIF4A.
Okay, wait, let's break down that mechanism. What is an RNA helicase?
A helicase is an enzyme that physically unwinds RNA molecules so that ribosomes can read them and build proteins. You can think of EIF4A as the motor that allows the cell to read the blueprints for building strong synaptic connections.
Okay, so it's the construction motor.
Exactly.
Yeah.
When cortisol artificially spikes the levels of BCYRN1, that 5' ALU domain acts like a wrench thrown directly into EIF4A motor. It jams the helicase.
And because the motor is jammed,
the local ribosomes cannot translate the messenger RNA required to build and maintain the synapses.
so it is actively halting the construction of the neural network.
Precisely. The data showed a significant reduction in the translation of genes responsible for extending axons and building synapses, most notably a sharp decline in a critical structural protein called PSD95.
What happens to a neural network if it lacks PSD95?
Well, PSD95 is the foundational scaffolding for excitatory synapses. It anchors the receptors that allow neurons to communicate. Without it, the physical connections between the neurons weaken, deteriorate, and basically fail to mature.
So what does this all mean? Are you telling me that a piece of ancient evolutionary baggage is the exact molecular switch that causes prenatal stress to physically damage brain synapses?
Yes. And what's fascinating here is that this specific piece of code, BCYRN1, is entirely primate-specific. You will not find it in mice.
Wait, really?
Really? This highly specific vulnerability only exists in primates and humans. It completely validates why modeling these diseases in human organoids is so vital.
Because animal models literally don't have the hardware to show this damage.
Exactly.
But how did they prove BCYRN1 was the exact culprit and not just the overall toxicity of the cortisol?
They use a genetic tool called HRNA, or short hairpin RNA. You can visualize it as a highly programmable pair of molecular scissors. They engineered these scissors to specifically hunt down and destroy the BCYRN1 transcripts within the cells.
So they proactively cut out the junk DNA before it can jam the motor. And then what? They flooded it with cortisol again.
Yes. And the structural integrity was completely rescued. The ribosomes were able to translate the blueprints, and the PSD95 scaffolding proteins were saved. The neural connections survived the stress exposure.
That is a staggering piece of molecular detective work. I mean, let's pull all of this together for you listening, because the relevance here is huge. We started with a soup of stem cells, perfectly timed their growth into an amygdala, wired them to a hypothalamus, exposed them to cortisol, and discovered a primate-specific vulnerability hiding in our DNA.
And clinically, we now have a tangible mechanism explaining how severe maternal stress during pregnancy might program long-lasting structural changes in a child's brain, potentially explaining the roots of anxiety and depression.
It proves how valuable synthesizing diverse fields can be.
Absolutely. This bridges embryology, genetics, and psychiatry. There's always more to learn when you combine perspectives like this.
It really makes you rethink everything. And I want to leave you listening with one final, lingering thought to mull over. This study proves that maternal cortisol can program fetal stress circuitry by awakening ancient primate-specific retrotransposons in our DNA. It makes you wonder, if our junk DNA holds dormant switches that shape our emotional architecture under stress, what other dormant retrotransposons are sitting deeply coiled inside our brains right now? What other ancient viral codes are just waiting to be triggered by environmental factors we haven't even begun to identify yet? Keep questioning and keep diving deep. We'll catch you next time.